Welcome back to the University of Illinois College of Medicine, the Microscopy Lab, and back to your STEM Microscopy with JUMP. I'm Shannon Egley. I'm the anatomy faculty here and anatomical instructor at JUMP Simulation. So now we're going to talk about Microscopy 201, animal tissues, and I figured it would be great to just stay in this lab because humans are animals too. So when we talk about animals, or sometimes it makes me a little bit, I want to correct people when they go, oh, these kind of animals and those kind of animals, but they don't refer to humans as animals, but we are. You know, insects are also parts of the animal kingdom. We're animals, bears are animals, dogs, your cat, we're all animals. So what's really cool is that when we look at these different types of tissues, collections of cells, if we're looking at us, or a dog, or a cat, or a bear, it doesn't matter because we're looking at very similar tissues. So I always thought that histology of the tissues was a pretty fun thing to teach in college because it's pretty simple, it's very straightforward, and it's one of those categories that's real easy to classify tissues, but people make it more complicated than it actually is. So we're going to try and make it as simple as possible. We're going to go to your manual. And the first page. Here you can see classification of animals, and like I just said, it's a lot wider than a lot of people think. Look, birds in it, reptiles, we're all in the animal kingdom. We're all part of the same group. Some of the things that make us like the others are that we all eat other organisms. So unlike plants that we talked about when we talked about cells, they're autotrophs. They make their own nutrient. Auto means automatic or self, and troph means to nurture, so self-nurturing, that's a plant. We're actually heterotrophs. We get our food from other sources. So we like to eat plants, or we eat other animals. We try and get our nutrients in ways that plants can't get. We can move freely. Plants are stuck. They're sessile creatures. They're terrestrial. They're stuck in the dirt of the earth. We have feet to let us move everywhere, which is nice. And we're also multicellular, just like plants. So lots of cells trying to control things, get us around to hunt our food, run from our predators, try and do things that plants can't do. Multicellular is kind of interesting. We'll talk about other creatures that are one cell that can move around, like yeast, which is a fungus. But for now, we're going to talk about animals. When we group them, we talked about this in the dissection labs that we do for under the sea creatures and the sharks, but it's how our bodies grow, really. So are we like coral that has no defined limbs? There's really no structure to us. We just keep growing this big wad or ball. Do we have bilateral symmetries, which means that if you split us in half, our left side and our right side actually look the same? Do we have a radial symmetry, like a starfish, where it has all these arms branching out in different directions? So we can be classified like that. And then how do we grow? Do we grow continuously like us, where we just keep growing all through our life, we don't shed our skin, or do we molt like other animals, like an insect that has to shed its shell? You've probably seen, if you're from the Midwest, like where we're making this, you've seen locust shells on trees because that locust gets so big inside of its little body and it can't grow its exoskeleton anymore, so it sheds the old one and has a new one ready to wrap itself up with. So there are a lot of ways we can classify animals. When we talk about the tissues, a cell is a cell. That's cytology. Cyto is cell, ology is the study of, right? Histology, or hist, is referring to a tissue. Ology is the study of tissues. What a tissue is a group of cells working together to get a common function done. So if you look at a cell that's really good at sending lots of signals, it might be a neuron. It sends signals from your brain to another part of your body. But if you see a chain of these working together, or a group of these working together, like in the brain or the spinal cord, because they're all the same cells working for the same function to send signals, we call that a tissue. It doesn't have to all be the same cell. It doesn't have to all be neurons. In fact, your brain's made up of a lot of cells that aren't neurons, that aren't that primary nervous cell that we think of. When we talk about plants in the whole plant chapter, it's the same idea. There are plant tissues that are all the same type of cell, but a lot of plant tissues are different types of cells working for one function. Is it to move water in the plant or food to the plant? Is it for protection? And we're going to find that with animals too. You can have the brain, which has a lot of different cells, nervous tissue in the brain, that a lot of different cells working for a common function. You can have the same thing when you look at skin, and we're going to do that. All right, let's go to the next page. Actually, let's look at some tissues. We'll go back. So we'll get this started the right way. Let's look at a tissue that a lot of people don't think is a tissue. I was just thinking about this earlier. Number 21. So in your case, don't forget, it says top. Open it that way. Number 21. A lot of people just think this is a liquid or a fluid, and it doesn't actually do anything as a tissue. We know that it carries nutrients. Wow, I'm getting really good at this. My numbering's off but just a little bit, but I still grab the right ones. So this fluid we think of as being transporting oxygen or waste products or nutrients around your body, but it's actually a type of tissue, and it's called connective tissue. So let's look at a blood smear just to get our skills all warmed up. So again, you're going to take your number 21, take your stage, lower it all the way down. I'm sorry, you can't see my stage. Mine's all the way down. I'm gonna make sure that my red objective, so that would be my number 4, is the one facing down. What do we call that one again? It starts with an S. Scanning. I'm gonna take my thumbs, put my thumbs down on the stage clips, holding them up. I guess I can lift this up a little bit. Stage clips, holding it up. And I'm gonna take my slide and pull it back. Put it into place. I have my number 1 light on, which is my underlighting. And if you didn't see it with the naked eye, right in the middle, you have a little reddish pink patch. That's what your target is. So we're going to put that in, and we're going to get that reddish pink patch right over our light source, so it glows. Looks like I'm all set. Let's look at our scope. Oops, scope is off again. Gotta fix that. That's not what I'm looking at. That's what I'm looking at. Okay. All the way down. I slowly bring it up. I'm turning the coarse, the fat adjustment knob. Oh yeah, already. Alright. So that's the line of that pink patch. When I do a blood smear, I don't get it square and perfect like that. They must slice it off with something. Like make it into a gel sheet or something and slice it. You may think that's blurry and you're looking at the wrong thing, but you're actually looking at the right thing. Okay. So I'm looking at that square patch. I'm going to try and get it as clear as I can. Yeah, it's less than perfect. In fact, it's a little bit dirty. So what I'm going to do is pull this out. I'm not as picky with my slides as I am with lenses, because lenses are magnifying. They magnify anything that I clean it with. So I'm actually going to take a chem wipe, a little tissue, drag across, Yeah, it looks pretty clear. If the prints and the dirt weren't coming off, I would take a little bit of eyeglass cleaner that you can buy at the dollar store for a squirt bottle for $1, or go to like a Walmart, go to their lens center, the glasses center, and just buy a nice bottle of squirt eyeglass cleaner. Yeah, that looks great. You can see my fingerprints over here, but not on my slide again. So let's slide it back in. Swoop. Oh yeah. Right, and there's the edge of my cover slip. On the right track for sure. There it is. Yep, that square patch is actually what I'm looking for. I'm in. Let's zoom in and see if we can see that a little bit better. Great, yeah. A lot of the specks are already gone, and so is my image. Let me bring that back up. Okay, perfect. All right, so I'm about as clear as I think I'll get. I'm going to take my revolving nose piece. So I'm grabbing the revolving nose piece. I'm going off the 4x, which is 40 times total magnification, remember? I know, isn't it annoying that I keep saying that over and over again, but some point, you're going to think it before I can actually say it out loud, and that's when I know that you're learning really well. Okay, wow, it just seems like a big pink blur, right? So what knob am I going to use now? I'm on the 10x objective, so 100 total magnification. There we go. I use my fine adjustment knob, and you can see it that pink isn't just a sheet. It's kind of like when we looked at the pictures in 101, where a picture from a distance looks like a picture, but you come in close and you actually see all those little tiny specks. It's exactly the same thing. So we're bringing this in. Looks really good. I'm going to blow this up a little bit. Nice. And I can see some little purple cells. I'm really interested in seeing some of the purple. So you're going to have millions and millions of these little red ones, but the purple ones, and I'm not sure yet. I'm thinking that's a flaw. Looks like a purple one there, some other little purple ones. I really want to find the purple, so I'm going to bring some of those purples. Oh yeah, there are a couple more right there. In fact, there's a double purple. I don't know if you can see it. So right in that area. Oh, right up there. Right up in here. Oh, there's one. Double purple. Let's see if I can get that closer. Oop, to the center. Alright, ooh, a couple double purples. Purple. There's a double. Okay, so now I'm going off my 100 total magnification. I'm going to flip it over to, there we go, by 40. So 40 times 10, 400. Oh. Fine tune only. There they are. So I might have a little bit of a dirty lens. Alright, a dirty lens. I'm going to bring this back. I'm going to use a piece of lens paper. So we just happen to have in the lab big sheet of lens paper. I don't need a full sheet, I just need the edge. Someone had ripped the rest of the page off. I can use that. I'm going to come down. And this is my 40x. I'm just going to swipe it towards me a couple times, see if that helps at all. So while I'm in here, I may as well wipe the iris. Alright, let's see if we can go back. Actually, I saw something interesting. Up here in the top, left corner, right here. See it? That's a really big purpley cell. I want to see if I can find that. So I'm going to move. Oh, went the wrong way. Nuts. There we go. Down, a little over. Ooh, there's another one right there. Straight over to the left. That looks like one that's really good to check out. See right here? Boop. But let's look at this one first. Right, I'm going to flip my objective. It's kind of like playing Where's Waldo when you're looking at red blood cells, so I have to admit, because they're so small. Red blood cells are hundreds of thousands of an inch. Whoop! I just messed up my whole camera. They're about 7 to 8 micrometers. Microns. Oh boy. Hope I can get my image back. Oh yeah, there we go. Great. Boom. Oh, it was there. And gone. In there. And gone. I still have some stuff on my slide. see that purple cell at the top. I see one down here at the bottom. Whoa, there it was. There was the big one. Bob, big old boy. Sorry. Wow. So if that had a better nucleus, I'd say that would be a megakaryocyte. So mega meaning gigantic, karyo, remember the nut or the nucleus, cyte means cell, megakaryocyte. In other words, reading it backwards, it's a cell with a nucleus that's big, megakaryocyte. It's really good sometimes to be able to jot down things like that, right? So if I were making a little note, I'd say, here's blood at my magnification, which was, I have the 40 lens times the 10 ocular, 400x total magnification. Blood, it's a connective tissue. I lost my image, let's get it back. And I can see lots of these blood cells. Look at the little red blood cells. I'm not going to draw every one of these, that's crazy. But if I draw a couple of them, the red blood cells look like hollow. So I'm not coloring them in. And this is as high as you can get with the 10x magnification from the ocular lens. But if you can get your 20 in and do the same thing, you have 800 times magnification, so twice as close. You'll actually see that all these ones that look like they're solid are actually kind of caved in just like this, because a red blood cell is kind of, if I sliced across it, it's bent here, bent here. Whoop. It's biconcave. It caves in here, biconcave. And we'll be putting together a whole blood stem too. So if I looked at it from the top, I see this kind of donut shape. What it actually is it's really thin here, thin, and thicker on the outside edges. There's no nucleus. So a red blood cell, by the way, we call them RBCs for red blood cell. They have no nucleus. My handwriting's terrible. Doctor's handwriting. No nucleus. So that means they don't have any DNA. So when you hear about crime scenes and they say, we collected blood at the scene of the crime, did they look at a red blood cell? And we got DNA from it. Did they look at a red blood cell? No way. No DNA. What happens to a red blood cell when it's formed is that it loses its DNA, it loses its nucleus, so it has more space for carrying oxygen. So it carried oxygen and CO2, carbon dioxide. They're about, this little squiggly means about 7 microns. And micron's written really weird. It looks like the letter U, whoop, kind of a long leg, and micron. Why is that important? There aren't a lot of cells I'll tell you the size of, but this is important because it's actually a measuring stick. So if I know that one of these is 7 microns and I'm looking at something that has 10 of these across, it's 10 wide, that's like putting 10 little rulers up against it. I know that whatever these 10 things are in is 70 microns wide. So it's really important. Right, so that's red blood cells. The other thing is a white blood cell, WBC. And their color, I'm going to say this, are purple because they're stained purple. They stain purple. Red blood cells don't. White blood cells stain purple. And they're going to have lots of weird shapes. Like I can have this gigantic megakaryocyte. Megakaryocyte. They don't hyphenate it. I'm doing this for you. That means big. Nucleus, or nut, is actually what it means. cell. So we read things backwards. It's a cell with a nut that's big. Megakaryocyte. Right? Okay, well, think of another good one. Okay, here we go. I've got another one. Let's say it's this big. Because in comparison to this mega gigantic karyocyte, I've got another one that might have a nucleus that looks like a kidney bean. This one's called a macrophage. P-H-A-G-E. Macro means big, so this is technically giant, but macro means big. Phage means eat. So this one eats big things, and that's what it does. This macrophage goes through your body looking for bacteria or things that don't belong, and it tries to eat it. When we talk about blood, we'll talk about that better later. And I didn't necessarily see, but we can check again. I'm not able to get this clear. If I could put my 20 time objective in, I would totally be able to see more clearly. That one stains dark all across. Let's see if I can move around and find even one. Ooh, look at that, there it is, right there. I get so excited sometimes, sorry. Let me find this. Can you see it? There's one that has a weird shaped nucleus. Right there. So let's zoom in on that again. See it? Let me draw it for you. So that is kind of kidney bean shaped. This one is kind of round. It actually has, it looks almost like there's a lump there, almost kidney shaped until I look a little bit closer, kind of a lump here, and another one. Okay, these we call polymorpho nucleocytes. Nucleosite. Poly means many. Morpho means shape, if you remember when we talked about bacteria. Nucleo means, well, nucleus. Site means cell. This is a cell with a nucleus that's shaped in many ways. So they have really oddball shapes. And that one is most likely a neutrophil. So it's kind of fun. When we look at tissues, you can name the different parts of it. We call this a tissue because it's different types of cells working together for one common goal. There are more than one red blood cell, but we're just red blood cells. We just call them red blood cells. But we have red blood cells and white blood cells, and we have a fluid that's called plasma. It's all in between. Plasma is normally like a yellowy straw color, but here it just looks white and clear. So that's kind of your introduction to tissues. We're going to talk about the 4 major tissues that are in the body, and then do a little fun thing at the end. All right, 4 major tissues coming up.